So there are sort of two main strategies to approaching science from the sort of experimental side or doing research side. And it takes primarily two forms. And I'm going to use the pen here. So you have sort of science approaches, and they're broken into sort of two broad methods. Discovery-based science, which we're going to talk about here, and I'm just going to use abbreviations for SPACE. And then the other kind is hypothesis-based science. So there are two different approaches, but of course, there's also going to be some exchange or overlap in them. And so we'll start here with discovery-based science. Discovery-based science is science where there's no hypothesis. It's really just observational. So the idea is you're just going to go in and you're going to observe and record what you see. And this is most commonly seen in environmental type sciences or ecosystem science. So an ecologist or someone like that is just going to go in and they might just watch the gorillas. They're like, okay, I'm just going to go in and watch what these gorillas do. I'll record what they do. I'll take pictures. The goal is really just to gather information. Not to go in and say, well, I want to manipulate what the gorillas do. I think that the gorillas will do this, and I'm going to manipulate them to do that. That would be hypothesis-based science. If we were to look in a different field, maybe like in cancer biology, discovery-based science, the approach there would be to say, well, I don't know what drug is going to work. I have no idea which drug is going to work. I'm not guessing which drug is going to work. I'm not making a hypothesis about which drug is going to work. I'm just going to try 100 different drugs. And if none of them work, none of them work. If 5 of them work, great, I'll keep testing those. But I'm just going to go in and look at random drugs and see what happens. So discovery-based science is also sometimes thought of as observational science. And if you think about that word, it's going to connect to hypothesis testing. So in many cases, discovery-based science will feed into hypothesis-based science or the scientific method. So over the course of the semester, we're going to do a lot of hypothesis testing, or again, this hypothesis-based science. Also in the old days, especially known as the scientific method. And so we're going to break the scientific method or hypothesis testing into 5 basic steps. The first is that there's observations made. So there's some sort of observations or information. This is that educated part of a hypothesis about something in the natural world or events or behavior or whatever it is. There's just some background information is also how it's thought of. And those observations are going to become the basis of a hypothesis. So you're going to try and explain those observations by forming a hypothesis. Remember, a hypothesis needs to be very specific. The reason why a hypothesis needs to be very specific is because it needs to be testable. And so when you make a hypothesis specific, that allows you to test it in a simple experiment. So you're going to form predictions from your hypothesis. If you can't make predictions from your hypothesis, and again, sort of thinking ahead to the labs, then your hypothesis is not specific enough. So you need to go back and reform your hypothesis to make it testable and specific. I think the other important thing here about hypothesis being testable is this is also the limits of science. Science cannot test everything. We don't have the experiments to test it or the interest to test it or the ability to test it. And so, you know, if you think about something like love, we can't test love. There's no way to test love. We can test sexual attraction. There's simple experiments, as you can imagine, to test sexual attraction. But we can't test love. So love is not within the realm of science. And so there are many, many things that are outside the realms of science because they are not testable. So just keep that in mind. All right, so the next step in the scientific method or hypothesis-based science is experimentation. And so this should usually flow pretty easily from your predictions. Your predictions should basically lead you to the kind of experiment you're going to run. So you'll run your experiment. In a good experiment, you'll have 2 sort of samples. And again, we're going to look at examples of this, so just bear with me for now in the explanation. You'll have your control sample and an experimental sample. And those 2 samples should be the same except for one thing that you're changing, right? So you only ever want to change one thing in an experiment. And that is what is called the independent variable, the thing that you're changing between your experimental sample and your control sample. And then once you've run your experiment, you're of course going to look at your data and you'll need to analyze your data. And we won't really do this very often in class, only one time in the semester, but scientists need to look for statistical differences. So, you can imagine that when you're running an experiment, sometimes you really want to see a difference. Like if you're testing a cancer drug, you really want to see a big difference. If you're, on the other hand, looking to see if something causes cancer, then you're hoping to not see a difference, right? Depending on your perspective. But the point being that we all have a perspective, and we either want to see a difference or we don't want to see a difference based on, our bias. And so to avoid bias, we have to use something impartial to tell if there's a difference, and that's statistics. And statistics is basically mathematical manipulations. Those of you heading on to nursing school will have to take a whole class about probability and statistics. But for our intents and purposes, just be aware that that is something that is necessary in true scientific experiments. And then based on whether there is statistically significant differences, we either accept or reject our hypothesis, or we support, this is another term we can use, we either support or reject our hypothesis. So all those definitions are great, but let's walk through a simple example to really see how this works. So I'm going to be writing with the pen again, so kind of bear with me. All right, so our observation, so the first step is that our flashlight doesn't work. So that's our observation. So we want to come up with a hypothesis that explains that observation. And so there's a lot of different things that could be going on, right? Your batteries could be dead, the bulb could be dead. That's just a couple of the possibilities. But remember that a hypothesis needs to be specific. So I need to choose one explanation. So I'm going to go ahead and choose that my batteries are dead. So my flashlight is not working because the batteries are dead. So notice that as I said that, it was a because. So remember, a hypothesis is an explanation. So the flashlight is not working because batteries are not, oh, whoops, batteries are dead. Sorry about that. So again, just emphasizing that hypothesis is explaining your observation. So let's make a prediction now. So remember, if my hypothesis is specific enough, I should be able to form a prediction that will lead me to a simple experiment. So my prediction then is that if I replace the batteries, then the flashlight will work. Right? Simple enough. So that prediction leads me directly to my experiment. So this is a pretty simple experiment. If only it were that easy to do research. You're going to replace with new batteries. Okay. So in this experiment, the batteries become the independent variable. You have your old batteries, you have your new batteries, right? That's what you're changing between your control sample and your new sample. Okay, so I'm just going to switch slides here to give me some more room. So I've done my experiment, I've replaced with new batteries. And so then I need to do my data analysis. Whoops, get back to pen here. So my fourth step is to look at the data. And there's two possibilities here. After I replace the battery, the flashlight works or it still doesn't work. Okay, And so based on those two possibilities as my outcome, I'll either reject or support my hypothesis. So if the flashlight's now working after replacing my batteries, then this supports my hypothesis. Okay. Now again, notice I didn't say the word proved, right? It supports that the batteries are dead. And so my question for you is there another reason why my flashlight would now work when I replace the batteries? So think about that for a second. What else would have given me the same result? And so, if you think about that, is I could have had my batteries in wrong. So maybe the batteries were just in wrong. And so, if I were to continue with this experiment, I'd want to go and test those batteries and see if they actually have charge in them. And that would be another experiment that I could do to continue to support my hypothesis. So notice that you're not really done just because you supported your hypothesis, right? And again, thinking about the labs this semester, you should always be asking yourself, what could I do next to continue to support my hypothesis? Because one experiment is not enough usually. All right, so the other possibility is that my flashlight is still not working. And so in this case, I would reject my hypothesis. Now again, does this absolutely prove that the batteries were not dead? No, right? It could be that the batteries that I put in that I thought were new are actually not, and they're also dead. So I would need to continue. I would need to continue these experiments. So when we support and reject the hypothesis, it's not the end of the story. It's really just a next step in a whole line of experiments that I might do. So that's a really simple example. Now we'll look at a more complex example that comes from your textbook, and this is one that's actually an observation from the natural world. So the observation here is that the leaves on maple trees fall in autumn when the days get colder and shorter. And so there's two things going on there. They're getting colder and shorter in the autumn. And so, now we want to explain this natural phenomenon. And so remembering that the hypothesis needs to be specific. So we need to pick one possible explanation, even though there's multiple things going on. The temperature is getting colder, the days are getting shorter. Those are both happening. But I need to make a hypothesis about one of them because I can only test one at a time. So in the example, they're making the hypothesis that the shorter amount of daylight is the cause of leaves falling. Okay. So again, if we connect this to a prediction, so from that hypothesis, I can make a prediction that if I take two trees and give them less light, then the leaves will fall. Okay, so in this experiment, remember that we need to make sure that the only difference between the trees is the amount of daylight. So the way they're going to set up this experiment is to have two greenhouses, right? The same exact type of tree in both greenhouses, the same exact temperature, the same amount of water, the same amount of fertilizer, the same age of tree. Everything should be as similar between these two samples as possible, these two groups as possible. So the control group is my group that again, is going to match my experimental group, but is going to get the same amount of daylight every single day. My experimental group is going to be exactly the same as my control group, except for my one variable. And remember, this is the independent variable. So let me write that here. This is the one that you change as the experimenter. So intentionally changed by experimenter. So in this case, that's light. Okay. And so also notice that the amount of light is in the hypothesis. So the independent variable should be mentioned in your hypothesis, okay? So make sure you're making those connections. Again, we're going to do this lots of times in almost every single lab over the course of the semester, identifying those variables. Okay, so they have the control group, they have the experimental group, they're going to let them grow, they're going to change the amount of light, slowly reduce the amount of light in the experimental group, and then they're going to measure the dependent variable. So the dependent variable, let me get down here, is the outcome measured. And so in this case, it's the number of fallen leaves. And I'll just point out that, again, the dependent variable should make an appearance in the hypothesis as well. So the shorter amount of daylight causes leaves to fall. So notice that the dependent variable is also in the hypothesis. Okay, so those are the two key variables in an experiment. There is also control variables. Control variables are everything that should be the same between both groups. So notice, and this is where some people get confused, you have a control group and control variables. Different thing. Control group versus experimental group. Control variables versus, whoops, sorry about that, versus other variables. Okay, so in this example, control variables include things like I mentioned earlier, should be the exact same type of tree, should be the exact age, should be the exact same amount of water, fertilizer, temperature, and so on and so on. Everything you can think of basically should be control variables between here. And they specifically mentioned time here. Time is often a control variable in almost every experiment. So 200 days for both of these. So the experiment's carried out, and that brings us to the data. And so here's the data presented for us. The control group lost this many leaves. The experimental group lost this many leaves. And they tell us here that a statistical analysis was done. Okay, and I just want to illustrate for you why a statistical analysis is so important. And so over in this corner here, let me again get my pen activated here. I'm just going to draw a graph for you. So here's my graph, and I'm going to draw this here. Whoops. So this would be my experimental group. This is my control group. So notice that between these two graphs, this suddenly looks like a smaller difference. This looks like a big difference. This looks like a smaller difference. But I can get to this from the same exact numbers. This is maybe 10 here, and this is 200, right? And the graph is on a scale of 200, right? So by having that graph be on that scale of 200, this looks like a big difference. If I make the graph be on a scale of 1,000, 10 and 200 are now a lot closer together, okay? And so I just want you to be aware of this. This is why statistics is important. And this is important for your everyday life. When you're watching those sort of shady ads on TV and they're showing you graphs and they're showing you data, it's very easy to make data look the way you want it to look by manipulating a graph. And again, that's why legitimate science is going to involve a statistical analysis, okay? So in this case, the statistical analysis was done. It shows that there's a significant difference, and so therefore the hypothesis is accepted. Okay? All right, so that's the scientific method. We're going to be using it a lot this semester, so make sure you're really solid on this and let me know if you have any questions.